Hello, welcome to what would have been Clinical Workshop 8 in San Antonio, Best Practices and Innovative Approaches to Clinical Research. My name is Alexandra Wilson from National Jewish Health in Denver, Colorado. My co-moderator is Laura Roth from Medical College of Wisconsin in Milwaukee, Wisconsin. Neither Laura nor I have any relationships to disclose related to this presentation. Our educational objectives are for research and registry coordinators to identify at least one quality improvement project that you can implement at your site over the next year, demonstrate unique practices to improve registry data entry, research awareness, and regulatory maintenance, and to evaluate past quality improvement projects and address ongoing improvement needs in a changing environment. These presentations will take us through the cycle of quality improvement principles related to clinical research. Topics include patient registry data entry, improving research awareness and engagement, preventing delegation of authority log errors, clinical research trial co-enrollment, and revisiting EQIP projects. Laura and I felt these QI presentations represented a part of each phase of the EQIP cycle. We'll start with how a site aims to improve research awareness, allowing sites to learn how to engage potential research participants. We'll hear how a registry coordinator has implemented timely CF registry data entry, making understanding your potential research participant population that much easier. Then a discussion of regulatory processes to prevent errors in delegation logs, a process that occurs at startup and throughout the life of a study. A large CF center addressed how to communicate between research groups in order to avoid unintentional participant co-enrollment in research protocols, maintaining data integrity, as well as equity among research protocols and participants. Finally, we'll end with a discussion from a site reflecting on past QI projects and how to move forward in an ever-changing research environment. Laura and I would like to thank our presenters for their time and effort, as well as the NACFC Planning Committee for having this online session in order for research and registry coordinators to showcase their unique quality improvement work. We look forward to these QI presentations and discussion after. Thank you.
Improving research awareness and engagement in a pediatric cystic fibrosis center. My name is Misty Thompson, and I will be presenting this topic today. Julia Meyer and Sydney Ross are my co-authors. We are with Indiana University, Riley Hospital for Children in Indianapolis, Indiana. There are no relationships to disclose related to this presentation. A little bit about me. I have worked in cystic fibrosis for a little over 8 years. In that, I have done a variety of different roles from working in clinic, and then past 5 years, I have worked in research, done a variety of different types of research and quality improvement projects. And then also I volunteer for the Cystic Fibrosis Foundation through fundraising and walks. A little background. Research is an important tool for improving the health and the quality of life with people with cystic fibrosis. Maintaining patient engagement in research requires active collaboration between patients and researchers. Currently, our center does not have a centralized list of patients interested in research. The purpose of this project was to determine the effectiveness of our current methods of research engagement and recruitment and find areas for improvement. Our current research interests in our current population, the best methods for contacting participants, raise awareness of research opportunities, and to develop a centralized list of CF patients interested in being involved in research. The methods used is we modified a CF clinical awareness survey to suit our needs. We distributed these surveys during just the in-person CF clinic visits between March and April of 2021. During this time, there were still many telehealth visits, So that's why we chose to do just the in-person CF clinic visits. We designed a REDCap database and entered the results. We wanted to simplify the surveys to make it just one page, just because a lot of times patients come into clinic have many other papers they have to fill out. So on the questions, we asked the relationship to the person with CF, the gender, how old is the person with cystic fibrosis? Have they ever received information about participating in a research study? Have they ever participated in a research study? If they would be willing to be approached for future research studies, what would make them more likely to participate in research? This was a multiple answer question. So the choices were early morning, late afternoon, or early evening. Those recommended by their CF doctor, non-drug studies, drug studies, research visits that only coincided with their clinic visits, those with not as many visits, and compensation for time and travel. The next question was also multiple answer. From which of these sources would they prefer to learn about opportunities to participate in research studies? A phone call, a text message, an email from their CF doctor, or from their CF research coordinator. 54 out of 57 of the surveys were returned completed, which was a 95% completion rate. It was equal number male and female subjects that filled out the surveys, and ages were anywhere between 0 to 18. For current research engagement, we have approached 83% of our subjects, and 17% have never been approached. Out of those that have participated, nearly 70% have participated in research, though 81% are still willing to participate in research. For those that have never participated in research, it was about 31%, but 11% of those are interested in participating in research. So there are a few areas of improvement that we can work on here. For study type preference, the number one choice was those from the CFMD recommended, which was 74%. The next two choices were those that coincided with clinic visits, which made sense. In our center, we're centrally located in Indiana, so sometimes the families have to drive for a few to several hours to come to clinic visits. Non-drug studies was also at 50%. The ones with fewer visits was only at 19%, which was a little bit surprising that they really didn't care. They had a fewer number of visits. For communication preference, the top two choices were from the CFMD to tell them about studies and to receive information via text. The two least common were from phone calls and from email. There were a few limitations to our surveys. The sample size was a little small. It was almost 20% of our CF patients. And then the surveys did not give a reason for why they didn't want to be approached. Was it because it wasn't good timing? Not right now. Is it because of the ongoing COVID pandemic? So in conclusion, we still do have a strong interest in patients wanting to participate in research. And clinicians play an important role in research engagement. For research staff, we may alter our communication with the data showing that text message is preferred over the emails or the phone calls. This is helpful information for study feasibility, since knowing what type of research is preferred among our patients would help with this. This also gives the possibility to have patient-tailored research. So our next steps is to redesign these surveys to include the reason for disinterest in participation in research. Distribute the surveys to all of the CF patients. Design a REDCap database to store this information. And then we can run reports tailored to the patient preference. We can distribute these to the CFMDs for upcoming studies and ultimately get the MDs more involved in research. Here's a list of my references. And we would like to acknowledge DB Sanders and Clement Wren for supporting us and encouraging us to always improve our practices and to do QI projects. Thank you.
Good afternoon. We encourage you to use the chat function to ask our speakers questions throughout the session. So we do have Misty live here to answer any questions you may have. The first question that we have, Misty, is do you feel like this project helped maintain a culture of research at your center, or that it helped to identify ways to improve upon the culture?
I believe that this actually helped to identify ways that we can improve upon the culture of research. We do have a pretty good culture of research already at our center, but I'm just trying to find ways to improve upon that and to get the message out just a little bit more to those that may not know about research.
Next question is, does your team feel like the survey helps with recruitment and enrollment in current and or future trials?
I don't believe that the survey actually helped yet with recruitment and enrollment. It just gave us a stepping stone so that we can design our database to help pull in those that are interested in particular types of studies, or to be able to send out messages via text or email, whatever way they prefer.
Okay. Speaking of text, what are strategies your research team can use to text potential participants in line with their institution's policies?
So we actually use REDCap for a lot of our studies and for some clinic lists. So for text messages, it is possible to do that now through REDCap.
Okay, well, that answers the next question about if you were to text patients regarding a study, would you use an app? So
Yes.
It looks like that is our last question for you. So if there's no additional questions, we'll move on to our next presentation from Sarah.
Hello. My name is Sarah Schwartz, and I am the Cystic Fibrosis Patient Registry Coordinator for the Pediatric and Adult Centers in Austin, Texas. Today, I will be presenting on timely data entry, specifically referencing our Pediatric CF Centers metrics, poster number 135. I have no relationships to disclose. Regarding data entry goals for the Cystic Fibrosis Patient Registry, our center's aim is to enter data within 30 days of the encounter date of service happening. By creating a systematic process with deliberate steps and surveying my progress along the way, I can hold my data reporting process accountable. In this presentation, I will discuss the strategies and tools that I use to track the throughput of my data reporting. Here's a little background for those who aren't intimately familiar. The Cystic Fibrosis Foundation manages and maintains the CF Patient Registry, nicknamed PORT-CF, but I just refer to it as PORT. Care centers collect patient health data and enter it into a database via a secure website. The website serves as an electronic data capture system, or EDC, with the ability to manage thousands of queryable data points. PORT has capabilities to run reports and queries with branching logic and can also generate patient summaries and other visual aids that can be shared with the patient and their care team. To enter data into PORT, you create what is called an encounter and you can specify the scope of data collected by choosing customizable tabs. All encounters that are started but unfinished will remain in incomplete status. The CF Foundation requests that all encounters be marked complete once the data has been entered fully. Both incomplete and complete encounters have their data filtered to CF Smart Reports, another website managed by the CF Foundation. CF Smart Reports is not a data capture system, but funnels the data already entered into Port and creates more reports and visual representations with which to manage care. Identifying trends in areas needing improvement helps create specific interventions for challenges such as BMI, lung function, and medication and prescription issues. If the data isn't entered into Port in a timely manner, then the data isn't in Port or CF Smart Reports to query. It is essential for a care team to have access to accurate, timely data to aid their quality improvement initiatives. And that is why it is important that you create an encounter in Port soon after the date of service has occurred, not months later, regardless of the encounter's complete versus incomplete status. Of course, the aim is to go back and finish entering the data within a few weeks of the encounter being started. At our centers in Austin, we utilize both paper and 2 separate electronic medical record systems. I refer to both of these as EMR/EHR and the patient's chart interchangeably. We also use a shared cloud drive to store and organize copies of patient data. I must access all of these systems daily to search for, identify, and gather the data to be submitted. We've got all kinds of incoming data that I break down into distinct classes: synchronous, asynchronous, and diagnostic results. Synchronous visits happen when the patient and care team engage in real time together. We've got clinical encounters like in-person and telehealth quarterly visits and associated activities such as respiratory cultures and lung function tests. Quarterly visits are scheduled and happen daily to weekly within our institution's facilities. There's also care episodes like hospital admissions at our institution, which can be scheduled or unanticipated. I access these data in the EMR/EHRs. Asynchronous visits include interactions between the patient and team, but not in real time. You've got a steady stream of communication that comes directly from the patients themselves, such as email, phone call, or text, which also gets notated and or uploaded into the chart. There's also outside correspondence from other entities involved in the patient's care, which can include encounter notes and supplemental interventions. These usually get faxed straight to the EHRs and then are filed into the patient's chart. Last but not least, we've got lab and imaging results, including both diagnostics that our providers ordered, as well as unanticipated results coming in. Most results get faxed to our EHR systems, and others are generated from orders in the EHR itself. However, not all results get filtered to the chart, so I must also log into extra third-party vendor application portals on a monthly basis to cast a wide net and capture results within 30 days of collection date. The data that I need to enter into Port is coming in for different patients at different times, constantly in the background. It ultimately settles into the chart, waiting to be discovered and reported. Some of the data I am aware of and expecting, and other data gets received silently, so to speak, and I am not aware of it until I happen to see it in the patient's chart or go looking for it purposefully. The system I developed depends on prioritizing specific data for various timelines. There will always be data I won't get to enter until months after it's happened. My power rests in optimizing what I can actually control. I do that by choosing to target certain data for same-day entry and save other data to circle back to later. Plainly speaking, I compromise and do a combination of real-time and delayed reporting. Specifically, I start quarterly visit encounters in port the same day they occur. Even if I don't finish entering all the data for that visit the same day, at least the encounter was started, albeit in incomplete status. It is imperative that I go back to complete these encounters and not let them sit unfinished for months. That's where my tools come in handy, so I can track which encounters I need to revisit and complete. It's time to talk about the secret sauce of my process and highlight the principal tools I use. I stay organized by the use of good old-fashioned spreadsheets on our shared cloud drive. One spreadsheet, the clinic prep sheet, serves as a catalog to track which data has been submitted to port. And the other sheet, the clinic flow sheet, acts as a data capture form. I personally created and managed the spreadsheet tracking tool that I nicknamed the prep sheet. The clinic prep sheet acts like a dashboard and tells me exactly where I'm at in the process of data entry for each patient. I use notation of my initials and date next to data points that I'm tracking, like a ledger. Without having to log in and navigate a website, and with one look, I can click on a spreadsheet tab and check the status of data I'm tracking instantly. Some of the main data trends that I continuously keep track of are quarterly visits, full data set, annual review, and team member consults. The clinic flow sheet is our data capture form and represents a single date of service for quarterly visits. It includes data points required to start an encounter in port, like date of service, location of visit, and team members consulted. I notate my initials and date in a column to record when I started the encounter in port. Usually 2 to 3 weeks go by before I have the time to go back, read all the chart notes, and finish adding the data into the encounter. After I enter all the data for the encounter and mark it as complete in port, then I notate my initials and date back on the clinic flow sheet with the date that the encounter was actually finalized. When it comes down to it, I'd say the most important aspects of my meticulous system are the use of the spreadsheets for tracking and reconciliation, and my strategy of starting quarterly visits the same day they occur. I follow the same steps every time I enter data into port, no exceptions. It takes dedication, time, and energy to maintain my system, but I see results. The data entry process as a whole that I've formulated consists of 6 steps: capturing the data, abstracting data from the chart, entering data into port via encounters, tracking submitted data in spreadsheets, reviewing tracked data in port and the spreadsheets, and lastly, reconciling verified data. And all the while, I am notating my initials and date in the spreadsheets as a way to timestamp completed steps within the data reporting process. Here is a flow map of my system. I acknowledge that there are barriers to following my process. It's tedious, among other things, but this is my checks and balances system that works for me, given the context of our institution. My main rule is I don't notate in my prep sheet until after I actually entered the data into port. If you see my initials and date next to a data point in the prep sheet, then you know and can trust it's already in the EDC. If you're not able to enter incoming data immediately, you need to track queued and pending data and verify that it's been completed properly. It is very easy to get overwhelmed and behind with hundreds of patients, not to mention staying on top of partial and final lab results and other unfolding events. If you don't stick to a system, then the care team won't have the data at their fingertips when they want to run a query for quality improvement. Now let's look at results. You can clearly see the difference from when I was hired to be the registry coordinator and applied my process in July 2016. Our pediatric centers baseline went from 40% to a sustained shift of over 70% in July 2016. Looking forward, our pediatric center will continue to enter quarterly visits into port the same day they happen. We will also continue to utilize our spreadsheets to keep an eye on incomplete versus complete encounters in port. I am currently working towards a new trend to sustain timely data entry above 80% within the calendar year. We are implementing the same process for our adult center as well. The information that researchers capture and enter into port helps us to better treat CF and improve care. Port is essential to a CF care center for managing populations, dynamic trends, and outcomes to provide the best care possible. Analysis and review of port data guides quality improvement for care centers across the nation. Timely data entry is paramount to keeping a care center on track. Here are my references, which are basically from official CF Foundation materials. If you have any additional questions regarding my presentation and process for timely data entry, please reach out to me via email. Thank you.
All right, we have Sarah here live to answer any questions. I do see one. Do you feel like your process has resulted in more accurate data entry for your center, in addition to the timely data entry?
I would say yes, because the process is requiring me to review things that I've already entered and I'm constantly reconciling. So I can actually catch my own mistakes that I make and correct them as I'm entering updated information. So I usually, rereading all the notes from all the providers, maybe more than once, just really making sure that I'm checking everything as I go down. So
I saw that there was obviously an increase in the timely data entry in the graph. Did you notice any in your center specific data, any drastic changes to kind of show that, it is more accurate?
I don't think we have any metrics to show that, except for in general, my capturing and being able to fulfill full data sets has been, I think, improving every year. That's the great thing about CF Smart Reports is it's showing year after year, and you can compare everything, every patient, every criteria point. So I would say maybe loosely, you can see some improvement, but not necessarily on every level.
One additional question says, very impressive. Any recommendations you have for other centers to apply your process?
I guess it really depends on your institution and what platforms y'all use. For Google, I just said the G word. If you have a shared drive platform, or if you're using paper, or if you have a better EMR, AHR that allows you to notate and have research participation in the EHR, our institution doesn't. But I would say the tediousness of going line by line and double checking if certain patients have everything in their chart is really worth it. I do it systematically. I try to do a sweep through everyone's chart and just to make sure, do I have all of this or that? And because I do that multiple times for different data points, I am getting a really thorough review of the data that I've entered. So yeah, it's I don't know, I guess I would say it's timely, but it takes a lot of time to be timely, but it's worth it.
And as one of the registry coordinators here, I totally understand. There's another question. Did your funding come up with the increase in your annual data?
I think so, because every year I've increased on both the peds and adult. I've increased the numbers of people consented and the number of full data sets that we've had. So I haven't really been on the ground level of completing the grants every year, but I'm pretty sure, just based on what CF Smart Reports is telling me, I'm assuming our centers have been getting more money every year.
Next question, is all the registry data input by just you or other people? Would you suggest for training others in this process?
It's just me. I'm doing both peds and adult. It's a fun little juggling project. I do have a few research volunteers that started working with me just this spring of like Marchish 2021. And they are IRB approved, and I have them double checking respiratory culture results and home spirometry. And then I also review each instance of their data entry. So I have a really close eye. So they are helping me a lot. And they can also do some of those chart sweeps I mentioned where I'm like, hey, go to every single patient chart and make sure you have this thing clicked on the annual review or something, and then notate as you go along and with their initials and date, and I can see their progress as they hit every single patient alphabetically. So mostly it's my show, but I do have a little bit of assistance, which has been really helpful. And I think it took like 3 months of training for the research volunteers to like really get it. The registry is a big website with a lot of stuff to look at. So I don't know if I have any good specific tips on training, but I guess being able to share your screen if you're not in the same place with the person you're training, like all 3 of my research volunteers, they're all remote in different cities in Texas. So I utilize, video chat, and I check in with them once a week and review everything that they do.
Awesome. Next question. It says, great presentation. Has your team considered moving your spreadsheets to Smart Sheets online, which can send alerts or reminders to you to complete an unfinished data entry?
I don't know. I use the incomplete center query report function in the registry. They do have a report where you can click on it and it shows you every single patient encounter that's still in incomplete status, and that really helps keep me on track, in addition to the fact that I am looking at my spreadsheets and seeing what I've completed and what is still outstanding. But I haven't thought about moving it to a different platform or whatever. I would have to ask people at my institution, if we can use this smart business you guys talked about. It's not Smart Reports. You said something else, right? Smart Sheets?
Smart Sheets.
Yeah, I don't have personal experience with it, but I know that our IRB, I think, was using it to track contracts for some things. So that's something I could look into.
Awesome. And I think you answered the next question. It says, do you only use your spreadsheet to track open queries on patients, or do you check your open encounters reports on 4CF? And I believe you said you do. I'm constantly checking everything, right? Smart Reports, the incomplete center report in the registry itself, my spreadsheets. So it's definitely, I've got lots of eyes everywhere. Right. Okay, I think that is the last question. So we will move on to our next presentation from Danielle.
Hello, my name is Danny Wolf, and I'm going to share with you today a QI project that Dr. Hugo Escobar and I worked on here at Children's Mercy Hospital in Kansas City, Missouri. It's called Plan to Prevent Delegation of Authority Log Errors. I have no relationships to disclose for this presentation. This is a copy of the poster abstract that was sent. We're going to discuss a little bit about this document right here in the center, and then this document down here in the corner. So the reason you might be asking, why DOA logs? That's certainly the question that I ask. It's really because we had a new research coordinator who was not only new to CF research, but new to research in general. And she was struggling with trying to delineate or delegate the task or assign the task on a DOA log that a sponsor had given her. She was starting up a new study. And in an effort to try and help provide clarity to her, we realized that DOA logs are really kind of a Rubik's Cube. They're a fluid document that require continual updates. They're not universal. There's no standard template that you can go to. They use different terminology and or groupings of tasks, especially when you're looking at different sponsors across the industry. And then the tasks can be so delineated that they may be overlooked. For example, we see a lot where there's vital signs and then pulse oximetry is listed separately. to us, we do pulse ox along with the vitals. And then tasks can overlap among study team members. And we really saw this more recently with the home coaching or the spirometry home coaching. We originally signed it to the RTs, but then as they got more busy, the research coordinators kind of had to take over that role. So it's kind of spread across the RTs and the RCs. An interesting thing when I mentioned about different terminology and our groupings of tasks, I looked at some previous delegation logs, and one of them had assess clinical investigation. And we were not really sure what that meant. I sent it out to the team, and we weren't really sure. We thought maybe it was the PI that should be delegated that task, but none of us really knew what it was for. And somebody said, maybe I'd get more clarification from the sponsor. So this is kind of what I mean by saying different terminology or different groupings of tasks. This is kind of a document. This is the one I highlighted first on the poster presentation, and we're going to discuss these 2 pages. This is actually a front to back document. It's a 1-page document, front to back. And we actually started with the second page or the back page, which is this side. And then we went in to develop page 1. And I'll go into a little more detail about that in the coming slides. So this is the page 1, and this is actually a good template for which the page 2, which is the reference guide, can help form. This is more for a study that is maybe investigator initiated that doesn't come with a delegated log. So then we have to make that up on our own. We can use this by looking at the template on page 2. And I'm going to discuss why we have both the lead study coordinator and the backup research coordinator sign this. So here's page 2. This is actually assignments of tasks. It's kind of groupings of tasks, similar or tasks that we do perform on a routine basis with a research study here at Children's Mercy. And then we've kind of listed out the team members that are typically assigned these duties. For example, we've got regulatory, this could be the startup, and this is like SIV, the delegation log. You could do the 1572. This is central IRB submission as well as our local IRB submission. So these are tasks really seen with the research coordinator. So we would assign anything in this grouping into the research coordinator's role. We also have the SIV. So this is something that the entire team does. So we would put everybody on this. We would have something like monitoring visits. This would be more the research coordinator and the principal investigator. So this is kind of how we set this as a guidance that can be done for page 1. Not only can this be used to create your own delegation log, it can be used to help to assign the task from when a sponsor provides a delegation log. Now I'm going to go into the chart review. So we decided as a team, it's not only important to have, guidance at the beginning of a study to set this up, and I'm going to touch upon that in here in just a minute, but we needed to do a review of the delegation logs at fixed intervals. For our team, we decided that January and July were the best times to do this. So the process is I send out an email to the team at the beginning of both January and July, and I say, all right, it's time to check delegation logs. And the team has until the 15th of that month to get back with me their review of their logs. We have this one document that is saved on OneDrive, and everybody fills this out for their active studies. So I'm just providing some examples. So we've got the study names in this column, the dates that the DOA logs were reviewed, the research coordinator responsible for those studies. Is the delegation log accurate? Yes or no? And if not, what needs to be fixed? So here's an example. We've got simplify study. Winfrey is the research coordinator. The delegation log is accurate, so there's no need to make any comments. We have a industry related study. So Vertex. Again, review date at the same time. B. Gates is the research coordinator. Was the delegation log accurate? No. And the reason is because he needed to add S jobs for MBW and remove F Nightingale as she retired. So he needed to put an end date on F Nightingale, and he needed to remove S jobs or add S jobs for MBW. And I will tell you, this is a specific example related to us. If this were to be reviewed and the research coordinator realized that another research coordinator had MBW certification and just needed to have that task delegated and signed off by the PI, that would not necessarily be an error. That's just something that's caught at review time. However, in this specific example of the MBW certification, we actually counted it as an error because it was a monitor that caught it. We did not catch it, so it was a monitor that caught it. Therefore, it ended up being an error. And then here's another example. Promise study. The date of review is July 5th by J. Bezos. Was the DOA log accurate? No. And the reason is because E. Musk is on DOA with MBW delegation, but is not certified. So that is truly an error. This did not happen to us, but I'm just saying that would be an example of an error. I think I mentioned that this is a OneDrive document so that we could all enter this information at the same time. After this information is entered on the 15th, Dr. Escobar and I look at this information and we review the findings. We look for information if there are consistent errors among the studies by different coordinators, then that's probably a teaching point. If they're just simple errors, then we just kind of move on from there. But we do share the results with the team in a weekly research meeting, and we review them. At one of our reviews, I was surprised by this. We did find out that some of the errors were made at study startup, which is why we initiated the lead study coordinator sign off on a document and the backup RC sign off on it. So we had to go back and what we did was a PDSA cycle to review our process to see what we could do to fix it. And we determined that we really needed to make sure that the delegation log was accurate at study startup. So in order to do that, we need the lead study coordinator to assign the task as they see fit and then have the backup RC review them and then discuss. If they're both in agreement, then that's great. If there's some questions, then it requires further discussion. But this is exactly why we developed this form for both the lead study coordinator and the backup RC to sign off on. We keep this with the delegation log in the binder. Now, here are our results. So we started this process where we were going to start the review in July of 2020. I'll be honest with you, I was quite surprised. I didn't think we had an issue with our DOA logs. I thought they were all perfect and we all knew how to fill them out. However, I was incorrect because of the number of studies we reviewed on the first go around, nearly half of them had errors. And I think this is due because, like so often, we get the DOA log, we do it at study startup, we file it away, and then we kind of forget about it unless there's something major that happens, like somebody leaves the institution. I think we get so busy with the study-related visits and the associated procedures and the processes and the documentation that this kind of takes a back seat. Maybe, we review it at monitoring time. So there was nobody more surprised than me to find out that we had nearly half of our studies reviewed have errors. This first go around, I believe the error rate was 44%. So I think it was 7 studies out of 16 had errors. So Dr. Escobar and I met over this, and then we shared the results with the team, and we decided we needed a goal. And the goal was going to be to reduce the amount of errors on the next review by 50%. So here comes the next review. It's January 21. We ended up reviewing 18 studies, and I believe only 4 of them had errors. So that dropped down to 18%. We met our goal because we had to reduce the goal by 22%, and we did meet our goal. We could have ended it there, but we all felt as a team that this was really a valuable process. DOA logs, they need continual monitoring and updating. And unless we pick a fixed interval or a periodic review time during the life of that study, we're not confident that we're getting that review in and making sure that the changes are made. So we decided to continue this process. With the next review, the most recent, which was July of this year, we had 17 studies reviewed and only one had an error. So we feel like we're on the right track. Also, as a team, we've adopted this process and it's actually been very well received, and we've all done very well with it. And it's kind of a commonplace thing now. After 3 reviews, we're getting more comfortable with it. So it's been, I would say, a success. In conclusion, delegation logs are fluid documents that require continual updates throughout the life of the study. Guidance via DOA template that standardizes the definitions and provides clear research rules is a helpful resource tool when assigning tasks to the appropriate team member. Also, using this document and combining it with a periodic review system, we were able to show a decrease in reported errors. I thank you for your time, and I'm happy to entertain any questions.
Great project, Danny. First, we just have a comment, not a question, and I couldn't agree with this more. It says, I love that you are not just catching errors, but identifying trends and learning opportunities as well. Well done. So the first question we have is, has this process been carried over to other logs, such as training logs and or other processes in the regulatory binder?
Not officially. We're starting new processes such as centralizing some of our IB training and protocol training, and that's become more of a centralized approach because as people are probably aware doing studies, that can get overwhelming too and get lost in the shuffle if you do training for every single protocol that uses the same IB. So we have another coordinator who has kind of spearheaded that, and we do that as a team where we do more of a centralized approach to that. So we're trying to streamline a lot of our processes.
Okay. Along that lines, someone asked, do you have a dedicated regulatory coordinator at your center, or do the research coordinators do all the regulatory?
No, the research coordinators do all the regulatory work to the studies that they're assigned as the lead coordinator.
Has your site ever created your own DOA templates instead of using sponsor provided versions? And if so, has there ever been any sponsor pushback?
We haven't, since this is still a fairly new project. We of course have done our own DOA templates for investigator initiated studies. Like I said in the presentation, I think we all kind of came into this new thing, or kind of surprised, thinking the DOA logs are not a problem. We'd filled them out for years, we understand. And then doing this process, we learned that really we do need some work, and there needs some attention to these. So we haven't used them yet. We haven't developed one like for a sponsor study. And I don't know if we will, honestly, but we have that guidance, and that's how we determine how the tasks are being delegated. I just recently received a study, and I have a backup research coordinator. We were going through the assignments of the delegation log, and even just between the two of us, we had a couple of questions on the assignments. And so we had a third person look at it, another research coordinator, and then we kind of all came to agreement. So it's still an ongoing process.
Next question. If the same studies are still open each time of your review, does that mean that the new studies are the ones with errors since you already completed the first review?
No, we mark those as done. I mean, we count them at that review and then they're done. So that could be, you know, why we do such a good job of decreasing our errors. But we also, a lot of us are very good at, once we make a mistake, we don't make it again. So when we kind of see this error on our log, we're double checking it the next time and we're making sure we don't have that error again. So.
All right, Alex, are there any more questions? Let me see one additional question. How long was the training period to learn and effectively use the new method you described?
Well, it did take a couple meetings and a couple months going through this. The first review was kind of a little sloppy, maybe. Our timelines weren't awesome, but we got much better as the second review came, approached, and we got it done with no issues. The summer one, this last summer, I think Dr. Escobar and we got it in at the very tail end, maybe, of our deadline, but we still got it in. So it's fluid. We make it work and we get it done and we get our 2 reviews in. So we're counting that as good.
Right. Okay, that looks like that's our last question. So we will move on to our next presentation from Katie.
Thank you.
Hi, I'm Katie Posch, and I'll be presenting on our QI project, Improvement of Clinical Research Trial Co-Enrollment, or TRACE. There are no relationships to disclose related to this presentation. To start, we're going to go back about a year and a half to the TDN Town Hall in July of 2020. During this meeting, Dr. Nichols presented slides on co-enrolling studies. The presentation discussed several ongoing and upcoming studies and the need to understand possible conflicts between studies when conflict isn't always obvious. He shared a study co-enrollment table similar to the one you see here, which has since been updated to this current version. This got our research team thinking. While we participate in 3 of these TDN studies and additional other treatment studies, we have a total of 39 open and enrolling studies. Currently, our research team is overseeing 79 studies. Evenly split between observational and interventional studies, they are in varying stages. About half are open, active, and enrolling. 6 studies are open and active, but are closed to further enrollment. These studies, while not enrolling, have ongoing study visits. Just over a quarter of our studies are completed but pending. These studies are closed to enrollment and visits are complete, but queries are still being resolved and the database remains open. 6 studies are listed as upcoming. These studies are in study startup with either contract, budget, or IRB approval pending before enrollment can begin. The remaining 6 studies are in the wings, which we call studies that are being looked at for feasibility or we are reviewing a solicitation. This study load makes the topic of co-enrollment very relevant to our research team. With all of these studies, we need to ensure that we have enough potential subjects. On average, 35% of our patient population participates in some type of research each year. After peaking in 2016 with a high of over 40% of our patient population participating, we started to see a decline in subjects participating in research. So an Equipped project in 2018 and 2019 worked on improving patient awareness of research and participation in clinical trials. Through the RC's efforts, a modest increase in new participants was seen over the previous year. This data was presented at NACFC last year. Thanks to these efforts, over the last 2 years, we are now able to see an increase of research participants again. This is great, but what does this actually look like? Are we getting more unique participants, or are we using the same subjects over and over again, potentially co-enrolling? Over the last 10 years, we have had a steady increase in overall patient population as reported in the registry. However, as our patient population grew, we did not see the same rate of increase in unique research participants. When we dig deeper into the study enrollments over the last 10 years, we see a yearly average of 12% of our CF population participated in an interventional trial. More than double that participated in an observational trial, and about 6% participated in both in the same year. Without digging up old subject logs and study binders, it's difficult to tease out whether some of the older studies had participants enrolling sequentially or concurrently. However, knowledge of recent studies suggests that many subjects have enrolled in multiple observational studies within a year, and that enrollment into both observational and interventional trials likely occurred when participation in one study ended. However, we can't be sure. Where has this brought us? That we know that the need to enroll subjects is continual. It requires the research coordinators to be impartial and follow sponsor protocols. This often leads us to enroll the same subject into multiple studies, either concurrently or sequentially. At the same time, research staff needs to be aware of these potential overlaps in enrollment, both for subject safety and for study validity. Communication between staff ensures best practices are being followed. With that said, I want to make a quick side note about our staffing and communication before going into the development of our co-enrollment project. Over the last decade, we have had 22 research coordinators work with our team. They stayed between 3 months and 29 years. Our current research team is on the newer side with a median of 3 years of experience. The 7 of us work in 3 different buildings on campus, in addition to our inpatient hospital located about 2 miles west of our main campus. These changes present many challenges in both team unity and study consistency, not to mention continual onboarding and training. That said, the research team came together and discussed the situation we found ourselves in regarding study enrollment and study participation. We asked ourselves these questions in regards to co-enrollment. How can we ensure all study protocols are being followed? How can we ensure equitable distribution of study participation? How can we keep data integrity between studies? And how can we best communicate between research coordinators? Taken all together, the number of studies we have open, the rotating nature of the RC position with varying experience levels, the number of patients we have, and the number who participate in research, we developed a project called TRACE, Improvement of Clinical Research Trial Co-Enrollment. Like any good QI project, we started by creating a global aim statement to help us focus and provide a starting and ending point. To highlight, the aim of this project was to improve our center's co-enrollment process with the goal of providing equitable and diverse research opportunities and reducing enrollment errors. To start, we conducted a systematic review of the research being done at our center. What studies are open? What studies are currently enrolling? Are they observational? Are they interventional? Next, we look deeper into these studies. What are the inclusion/exclusion for each study? Is co-enrollment okay? Does the protocol even mention co-enrollment? We had discussions with PIs and study sponsors if the answer wasn't obvious. Then we brainstormed the best way to track studies, the subjects and requirements of each, and have it accessible to all RCs and have it HIPAA compliant. At our center, our EMR isn't capable of this. We settled on the use of REDCap, both for its ease of use and confidentiality. We discussed what we should capture in REDCap without recreating study documents already available. Once built and approved, each research coordinator is delegated with updating the studies and the subjects they enroll. So what does this look like? Within REDCap, our project has 2 study arms. Arm 1 is subject information, and this arm has 2 forms, subject demographics and subject enrollment. For subject demographics, we try to keep the information simple. We collect name and initials, date of birth, registry ID, and any relevant notes the RC might deem important, such as this person lives out of state. The subject enrollment form asks for information about the study this person is participating in. It includes study name, study PI, the primary RC, the type of study, along with dates of enrollment and completion. Obviously, we also ask if the study allows for co-enrollment. When complete, a repeating table shows the studies this person is participating in or has participated in with dates and co-enrollment status. Arm 2 is information about each individual study. This arm also has 2 forms, study information and study subject list. For study information, we again collect study name, PI, primary RC, the type of study, IRB number, the study status, the length of the study, and enrollment window and goals. The subject list, which captures names, ID, and enrollment date, can then also be viewed as a repeating table. To date, 105 subjects' data has been captured. These 105 subjects account for 219 study enrollments, 197 into observational studies, and 22 into interventional. 51 subjects were enrolled in multiple studies, anywhere from 2 to 7 studies. The majority of subjects co-enrolled tend to be into observational, specimen collection, or single visit studies. These enrollments are into about half of our open and enrolling studies, 5 interventional and 12 observational. Of these, 2 do not allow for any co-enrollment, and another 2 have specific requirements about co-enrolling. There were some inconsistencies in data entry regarding study name and co-enrollment ability, and a significant lag time of the data entry of new study participants. However, we found no co-enrollment errors. The use of TRACE and the data we've analyzed from it so far show some successes and some challenges. Using REDCap, it is easy to use. It allows for multiple users at a single time, and it gives a quick snapshot of enrollment by subject or by study. However, the challenge is, it is an extra step in the screening process. There are some inherent limitations within the database we are using, and these inconsistencies in data entry can cause for some difficulties. So in conclusion, co-enrollment can allow for rapid completion of clinical research trials, but may cause issues with data integrity and subject safety. TRACE is a novel process for highlighting subjects and studies that can benefit from co-enrollment, while also identifying those subjects and studies that cannot. Acknowledging the potential advantage of co-enrollment while recognizing the possible risk to study outcomes will benefit future research trials and the people with CF that participate in them. With multiple trials open and more on the horizon, it's important to recognize the restrictions of co-enrollment on each study. This recognition ensures that research trials will provide quality data-driven outcomes, limit conflicting goals between studies, and provide people with CF equitable and diverse research opportunities. I would like to thank the group of research coordinators and PIs that I work with every day, the clinic and hospital staff that are always there and willing to help in any way they can, and especially to the people with CF that participate in all of our research studies. Thank you.
All right, we have Katie here to answer your questions. First question I have, excellent work. I love the REDCap database capturing patients and study status. Is the RC responsible for entering this data?
They are. So each RC will do their own studies. So pulling off of the last presentation about the delegation logs, the primary RC for the study will be able to, would go into the REDCap database and enter their study participants and what, if that study allows for co-enrollment or not.
Next question, is this a running list that can be used to keep track of potential research participants for the future?
Kind of. So right now, our goal to do it was to record the active participants that we had who were in active trials that we knew could or could not co-enroll. The goal eventually would be able to go back and enter some other lists. Once the patient's name is entered into the database, we don't go and delete them from the list. So if they participate in a study and they're added to this REDCap database, then they will stay there, but their status in a study participation will change. So if they're in a trial that lasts a year, you will see that they're in this trial for a year, and whether or not they could co-enroll during that period. And then once that study's end, it would still be kind of in their record.
Okay. Next question, are you able to print or otherwise see who has participated in the past so that research and clinical teams or even patients can track what one has participated in the past?
So this wouldn't be available to patients. We could, if someone was interested in like, oh, I did a trial 2 years ago, do you remember what that was? Again, the goal would be to eventually get some of the historic data in, but right now we're focusing on the current list, and we only have it accessible to the research coordinators, but just like any REDCap form, if a PI or other clinical team member wanted to see kind of what their study status looks like, what this patient has participated in, just the way that we built it in REDCap, we could pull out a report or print some lists if anybody was interested in that.
Okay, great. Next question, did you have to put this through your IRB? If so, were there any issues? Was consenting needed? We cannot use REDCap without IRB review.
We did not have to put this through our IRB because it is for our IRB, it's listed as a quality improvement project. We are all of the subjects that we're seeing and all the research coordinators as a team, we tried to limit as much of the PHI. I mean, yes, we have their names in there, but all the research coordinators are able to see the list. And since we can do QI through our REDCap, we are able to use it in the REDCap capacity.
Okay, great. So if someone wants to add to the research coordinator responsible question, they are asking if the data is entered following screening.
We usually do it after once they're consented into the study. So if they have not been, if they're screened just like as an initial blush to it, they would not be entered. But if they have consented into the study, then yes, we would recommend that they would be entered into the database so that we can track them so that we're not overlapping on study protocols.
Okay. That looks like that is the last question I'm seeing. So we will move on to our final presentation from Teresa.
Hi there, I'm Teresa Kump. I'm the research manager for the Pediatric CF Care Center in Milwaukee at the Medical College of Wisconsin. I'm grateful to have the opportunity to share with you what we've been focusing on for quality improvement over the last 18 months. We've been reflecting on past projects and addressing ongoing improvement needs in a changing environment. I have no relationship to disclose related to this presentation. So just to set the stage a little bit about what I'm going to be talking about, for us in Milwaukee, as I'm sure for many of you, 2020 to 2021 was a period of reflection and transition. We dealt with and addressed the impacts to research brought on by the COVID-19 pandemic. This included a pretty rapid, but fortunately temporary suspension of what they considered non-essential research activity. We had research coordinator furloughs, which of course redistributed work to other personnel. And then there was this kind of slow and steady reawakening of research, and that led us to kind of where we're at now in this hybrid work model where coordinators are working both remotely on some things and then in person for other activities. And for us in Milwaukee, we had another kind of significant change where our pulmonary clinic, where our CF patients are seen, relocated to a new clinic space on our campus. Brand new building, beautiful, but it has a lot of new processes and technology and staff and space and resources, all new. And we were the first inhabitants of this new clinic space. So there was kind of a lot to learn there and navigate while we're kind of going through all this in the last year. So in thinking about that kind of period of reflection, rather than embarking on a new equip project for the year, we decided to think about some of the past projects that we've done and kind of look at what those improvement changes were and reflect on them. It's the final step in our equip wheel, and we thought it was important to address that and kind of evaluate, did our projects stand the test of time? Were they amenable and adaptable to the new working environment? Did they work in our new clinical space with our new staff? So we just wanted to see where we were headed. So each year when we do think about our equip projects that we're going to do and set our goals, I like to think about them as kind of falling into two kind of separate categories. We have projects that we've done that focus on the culture of research, things like communication and collaboration, things that build your relationships with our research and clinical partners. And then there's sometimes that we really focus on some pretty concrete operational improvements that really tackle some of the challenges that we face in our research program. And of course, they are often very closely combined with each other and interdependent. So we targeted, looking back at the last 4 different projects that we've done to evaluate the continued success of them. In 2016, we audited our Port CF consent, and we found some quality gaps, and we really focused hard on getting a process in place that was going to eliminate those compliance gaps and ensure consistency across all the consenting. In 2017, we focused on integrating the CF clinical care team and the research team, really build that partnership and relationship there. In 2018, we were increasing the communication between our CF research team and our pulmonary clinic staff outside of the CF program. So the MAs, the nurses, and the front desk staff, all the people that we partner with for our research visits. And then in 2019, we were working to increase our collaboration between the pediatric and adult CF research teams. We are on the same campus. Sometimes there's overlap in the studies that we do, but we are at separate institutions, and we really wanted to build that relationship there. So looking first at the relationship between the research program and our new clinic, I think that the work we've done in prior years to increase the communication with our clinical staff and ensure that research is included in clinic operations was essential this last year when we moved to this new space. Because we had those pre-existing relationships, we were really able to establish a physical presence in clinic. This included just working with the team to make sure that there was a workroom in clinic for our coordinators. It's a storage room as well as a workroom, so there's space for a workstation, a counter for prepping kits and so forth, and a really nice storage area for all our million kits and boxes and so forth. So it's kind of become the envy of the new building. We also wanted to make sure that the coordinators had access to the technology systems that were being rolled out with this transition to the new space. There was initially not a plan not to have research coordinators access to this communication system, which is kind of replacing a paging system, but we were able to advocate for that, and the coordinators now communicate in that way. And we also wanted to make sure that the research activity in general was included in a new tracking board that they have in clinic, which manages the clinic flow of a patient, who's seeing the patient at which time and who's on deck to see them, make sure that research was included in that. So if a patient did need to see a research coordinator, they weren't going to leave until we were able to make sure we saw them. So that was really helpful. And I think having built on those predetermined relationships was really valuable this year. One of the things that kind of came out of that project we did that year to work with our pulmonary clinic staff was the development of this intake sheet. It kind of tracks where what a patient might need while they're in clinic for a research study visit, especially the special things they might need, like a second set of vitals, an extra culture, and so forth. So we developed this in 2018 in our old clinic, and it was kind of exciting to try it out with our new our new staff that's supporting us in the new clinic, a new process. Patients are roomed a little bit differently now, so it was really important that we introduce this and work with them, make sure it's still working, and continue to train and partner with the new staff in the clinic so we can continue to collect the information that we need for our research studies. So it was really reliable to be able to use this in our new space. The year that we really focused on integrating our CF clinical care team and the research team, I think has been, one of our most successful endeavors. That year, one of the biggest accomplishments I think that we had was when we review patients every week, the clinical care team reviews patients, who's coming in, who do they need to see? Do they need to see psychiatry? Do they need to see the dietitian or the social worker? We included research in that list. And that way, whenever we evaluated who was going to be coming in, we researched literally and figuratively had a seat at the table for that discussion. That was incredibly valuable during the especially the early and mid stages of the pandemic, when we really had to limit research coordinator presence on campus. So we could just really very carefully plan who was going to see which patients for research and make sure everybody knew what was going to be happening to that patient. We were, as a care team, able to very quickly pivot our weekly patient huddles, clinic meetings to Zoom. So we're kind of all on deck and continue to have those discussions. During that meeting, when the patient review happens, the CF nurse updates the list with who needs to see each patient, and she takes that information and makes sure that that gets translated to the tracking board that we have in clinic. So it was really nice to see kind of those two processes really complement each other to ensure our continued success. Focusing on the registry consent compliance, also we were pleased to have found it very sustainable and reliable during the pandemic period. Of course, even during a pandemic, it's important to maintain our regulatory compliance. This previous eCRIP project really focused on making sure there was consistency and reliability in who was obtaining consent and making sure that the consents were valid before the patient was being entered in the registry. This map here is kind of what happened before our project, where there was it was all over the place of who was maybe approaching the patient and getting consent and handing it off. And so we had lots of errors there. And what we did that year was really streamline this process. Consent is only obtained by a member of the research team, and it's handed directly to the registry coordinator to validate signatures and make sure everything is compliant before the patient is enrolled in the registry. We made some minor modifications during the remote work period, where if it wasn't, the registry coordinator wasn't on site during the day that this consent was occurring, we would scan the consent to her and she would validate those signatures before the patient was entered in the registry. This was able to be done for new patients being enrolled in the registry, as well as tracking patients for guardianship changes and age of majority and so forth. And we're really happy that we're able to continue to maintain this process without any errors of noncompliance. And then our final project that we wanted to reflect on was the work that we did to improve the collaboration between the pediatric and adult CF research teams. During the year that we worked on this, we developed a cloud-based file sharing system to transfer information from the pediatric to adult team, let them know what studies a particular patient might have participated on while they're on the pediatric side, and just kind of hand off that information to the adult team. So already having that process in place was just able to continue. It was obviously something that we can continue to do virtually and remotely. So that was really successful. And I think as a direct result of this strong bond that we made with our partners at the adult research team, we were able to increase referrals from the adult program to the pediatric program for research in the last year, particularly for the CHECK study. So we were really excited to see that just really result in those really measurable successes. And I think we learned, as again, reflecting on this, that sticking together was so important. You know, having those friendships that we forged and continue to have was just really, really important for us throughout this year. What we did before was develop these meet and eat lunches, of course, because everybody can lunch and learn. Sometimes you just want to get together and talk to your colleagues, share experiences. And so we continued those. We, of course, transferred to a virtual environment, did those by Zoom. This is a little picture that we did. We had an after hours wine and dine event with the research team, and that was really nice to just stay in touch and just really maintain that continued support and ongoing mentorship and collaboration. It was, kind of now more than ever, it was really valuable for us. So this is part of my kind of time of my talk. I usually say reflection, but since our whole year was on reflection, I guess I just wanted to end with some takeaways. I think what we learned is that quality improvement, we know this, but quality improvement doesn't end with the abstract. It's really meant to really help improve your program, focus on things that are going to make your workflow better, the research subject participation better. It's all about, continuing success. And to do that, it does require ongoing evaluation for sustainability. Times change, work environments change, space changes, people change, all those things change over time. So it was really important for us to just to evaluate that and see how they were going. And I think just fundamentally, I think that we kind of realized that our efforts to improve the culture of research have long-lasting and far-reaching benefits. And we really did feel that, and it was nice to reflect on that and see how far we've come. I would like to acknowledge and thank most especially our co-authors, Laura Roth, Danielle Groff, and Dr. Nicholas Antos, and of course the CF Care team and the Pediatric Pulmonary Division at Children's Wisconsin. Really appreciate being able to talk to you today. I so wish we were there in person, but hopefully next year, and to look forward to questions and discussion. Thank you.
Hi, Teresa. So we have one question so far for you. What are ways you think teams can choose QI projects that can be sustainable and applicable in the future?
Thank you. I think choosing a project, that's a really great question. So I'm just going to pause. I think the EQUIP program has a great tool to help centers narrow down what they maybe want their focus of their QI project to be. It's a nice place to start because, and again, it kind of helps you look at environmental things and resource things and staff and people things. So I think it's a great place to start. In terms of making sure things are sustainable, I think QI in general, when you do kind of the root cause analysis and you figure out how you're going to solve your problem or look at how to solve a problem, not focusing on one a person problem, right? Focusing, is it a resource problem? Is it environmental, or is it a process? All those different things, or maybe it's all of those things kind of put together. So things like that will help it then translate as times change. You might have to adapt your process a little bit, but it's a project in itself that you can continue to revisit rather than focusing on it just being an individual kind of specific or person that's contributing to certain things. So that's what we've kind of learned. And I do, like I kind of said before, I like to have projects that focus on the culture of research, and then ones that really work on these operational things. And like the year that we did the check-in visits, and it was about culture of research, and then we got this operational improvement out of it with the intake sheet. So I think that focusing it on those two different sort of avenues of QI, I think has been helpful for us. Okay, next question. For file sharing, are data use agreements required to share those, or is it within the same institution? It is all within the same institution because it is through MCW, our academic institution, is a cloud-based system that we use. Yeah, and we also were one CF center as well, so we share the registry. We have all of our all of our staff on both the adult and the pediatric side are members of the same research team for the registry. So that allows us to also collaborate on any kind of information related to that.
Okay. I am not seeing any additional questions. So I guess since there are no additional questions, we'll just end our workshop a little early. Alex and I would like to thank our speakers for sharing their quality improvement projects, and also thank our audience for attending our session. We hope that you are inspired by this session to work on some new QI projects at your site, or to maybe review some past projects. We invite you to virtually stop by our speakers posters, and hope you enjoy the rest of your conference. Thank you.